and kind of walk you through what I find so interesting about them. So algae are a polyphyletic group of organisms. They're in the Protista, and they encompass the group algae encompasses the diatoms, dinoflagellates, red algae, green algae, brown algae, including some of the oomycetes. So if you look at this tree, you can see that here's like the root of a tree, some ancient ancestor. And the diversity of life kind of radiates out. And a lot of the algae are super distinct from each other in the sense that like um a land plant is more closely related to green algae than brown algae is related to green algae. So it's really interesting. And things like dinoflagellates, which include the zooxanthellae that are symbiotic with corals. These are more closely related to like the malaria parasite than other algae. So they're photosynthetic protists, and we often lumped in a lot of the um, cyanobacteria as well, just because it's this photosynthetic uh, bacteria. Generally, when we're talking about the seaweeds, we're talking about the macroscopic algae that live in the ocean, and these are handily color coded. So the rhodophytes are the red algae, the chlorophytes are the green algae, and the phaeophytes um, are the brown algae. So the red and greens are over here, and the brown algae are over here. And the brown algae are going to be like your kelps and such, whereas the green and reds are a little bit more hard to identify quite often. When you go to the coastlines and the rocky shores or even the sandy shorelines and the more protected areas that have a lot of your rockweeds, your knotted agracts, there's going to be a lot of zonation, and that's going to dictate the height of the shoreline is going to dictate what kind of species you're going to see. And that's important when you're thinking about identification. And this book is really good in the sense that it's going to tell you what height the shoreline, or whether it's a subtidal species, you'll find the species most commonly. And that's a good tool for ruling out species that look quite similar to each other. So if we look at a rocky shore, you often have a splash zone up in the top of the rocky shore where you'll find a lot of limpets, periwinkles, and your pelvicia. I can't remember the common name of it. Very small rocky rockweed. Then you'll get to your high tide zone, which has a lot of barnacles, limpets again, more of your rack species like Fucus spiralis, Fucus vesiculosis, those kind of species. Then you'll get down to the mid tide zone, and you're going to get a little bit more diversity of seaweeds, maybe more reds and stuff. And the low tide zone is where you're going to start finding your kelps and things that live in the margins of the subtidal and the intertidal zone. On a kind of gently sloping shore, like for instance in Connemara, you will have a domination. You'll have your high tide zone kind of dominated by the pelvicia still, but knotted agrac's going to really take over that mid-level tide. And once you get down to subtidal or very low tide zone, you're going to get a little more diversity of brown seaweeds. But that sediment bottom is a little bit harder for things to settle on, especially sessile organisms like seaweeds. So not as diverse generally in those habitats. And that's generally, this is one of Tom's um, drone photos, but this is generally what you're going to see in that in like Connemara, in the gently sloping shoreline areas. Now, seaweeds are really cool in the sense that they have um, many variations in an alternation between a diploid and a haploid life cycle. And that kind of the alternation of those two stages within the life cycle is going to be characteristic of families and even phyla. So if we think about haploid and diploid, haploid means you have one set of chromosomes, diploid means you have two sets. If we look at humans, most of my body has, the cells in my body have two sets of chromosomes, but my reproductive cells, the eggs, only have one set. So when, let's say, mammals undergo sex, the sperm and the egg recombine and they create a zygote, which then has two sets of chromosomes. So that action actually is where we get a lot of our genetic diversity within a species or a population of a species. And we basically have just a very short time period where you only have one set of chromosomes, right? It's just the sperm and the egg stage. In a lot of macroalgae, they'll have a whole stage of a life cycle have one set of chromosomes, and a whole other stage have two set of chromosomes, and they'll be free living, not parasitic or embedded within the diploid organism. So here's an example of a green alga. This is ulva, and you have, let's say, your haploid stage, the gametophytes. They produce gametes, which is the same as a sperm or ovum. They then form a zygote and form the diploid stage, which is called a sporophyte. That sporophyte then undergoes meiosis, produces zoospores that then become the gametophytes. So you have these two, in this case, identical looking stages that have different reproductive functions in a sense. If we look at kelps, and this is really important for a lot of the research we are doing, there's a non-identical variation in the haploid and the diploid life cycle stages. So you have your sporophyte with two chromosome pairs producing zoospores through meiosis on the blade. Those then develop into microscopic gametophytes, so multicellular, but you can't see them in the environment, that then reproduce and produce the sporophyte once again. So this is kind of, it's called a heteromorphic life cycle. When you get to the red seaweeds, it's actually super, super complicated, and that's what makes aquaculture, well, both for the kelps and for the reds, so kind of complicated and not as cut and dry as you might expect. So you have the same thing where you have haploid and diploid stages, but you have a third stage here. It's a triphasic life history. So on the female gametophyte, once it's fertilized, it produces carpospores. It's called a carposporophyte. So you have a haploid individual with these diploid structures embedded on them. They can be very distinct in the wild, and it's going to be one of your tools if you get into the really niche kind of identification using microscopy with reds. It's going to be one of your tools for identification, how those structures are shaped and look under the microscope. So the carpospores generally form the tetrasporophyte, and those go on to produce gametophytes. So just three phases in there. And that's really important. Like I said, for aquaculture, this is an example of nori. So it's a very important species that is grown in Asia. You know, it makes seaweed wraps for sushi and such. So you have your macroscopic sporophyte here, then it or gametophyte, sorry, that produces carpospores once it's reproduced, and then it forms a filamentous conchocelis that only really grows on oyster shells, and kind of creating or identifying that life history cycle is really important for that aquaculture industry in the 1950s, and it continues to be really important for a proliferation of algal aquaculture around the world. All right, so again, your guidebook is probably this. It's very easy access. I've tried finding something similar for California, and we just don't really have that. Have this like nice picture guide with all the information in it. So this is really cool. And it's what most of the lecture is based on. But if you want to get more information into the diversity of seaweeds, I think there's over 350 seaweeds known for Ireland and Britain. You can look into these books. They're a little bit dated, but you can always go to what's called Algae Base for an update on the species descriptions and species names, because nomenclature can often change once we kind of identify differences in the range of species and so on. You know, we're just kind of building more and more information every day. All right, so now I'm going to talk about the morphology of seaweed, which is it's going to be the way that you identify stuff in the field. Generally a blade or a frond of a seaweed. It's going to be described like that. Kelps will have a stipe, and so will a lot of the reds, maybe not so many of the greens. And then all seaweed are going to have a holdfast. And the shape of the holdfast is going to be really important for identification, distinction of a lot of species. A lot of the browns will have air bladders or a nematocyst. Again, this is a frond. You can have what's called a midrib, which looks like a vein in the leaf. Very similar, analogous structure. Here would be like a claw-like holdfast. This would be like a discoid holdfast here. And then this is like an example of maybe a red alga, and you can see these dots here would be distinctive, and these would likely be a reproductive structure of some sort. The branching is going to be really important for your identification, again, especially in the reds, but also with some of the what used to be called cystosiren. I'm drawing a blank on its genre right now. But some of the browns is going to be really, really important for identification. So you have alternate branching here. You have distinctus or pinnate, where it looks more like a feather. Unilateral, it's only on one side. You can have spiral branching, whorled branching, and then decussate is a variation of whorled, but it's like alternate, so it's branching in one plane. Dichotomous branching means it forks at each junction. Trichotomous is a variation of that with 3 forks. And then cervicorn is a very unique kind of unilateral branching. Looks like this, and you're going to see this in the reds and the Plocaniae leaves a lot. We also have a few free-living species. So here you have your merle, and you might see this in parts of Connemara or further down the west coast. It can also be called a rhodolith, and that just generally indicates that it's not branching. It's kind of a free-living solid form. And then we have our crustose species, and this is generally going to be the reds, although there's a few brown crustose stages of species. So this is a crustose corallin. This is an example from a coral reef where it's covering a dead reef, but it basically is fits to the substrate that it's on here. But you can also have fleshy red algae that are crustose too. And the easy way to distinguish between those two when you're dieting or when you're in the tidal is just to feel the texture. And texture is really important for a lot of these species as well. All right, so I'm going to start with the reds, and I'm going to start with the corallins because these are very distinct in this area. So when we talk about the thalli, corallins are unique in that they're sections of the thalli, and they are called the intergeniculum. These are the apparent sections, and the sort of elastic connections between the intergeniculum is called the geniculum. The shape of this and organization is going to help you identify your species. So Corallina officinalis, very common. It can form turfs in the intertidal that are very diverse with smaller invertebrates. You can see it has this pinnate branching here with very small intergeniculum. And then you have Corallina cespitosa. It also has pinnate branching, but the shape of the intergenicula is going to be very different here. And then if you look at Ellislandia elongata, again, the shape of the intergenicula is very important, but again, pinnate branching. So you really have to kind of focus in on the structures there. They could be described in the very same way, pinnate branching, forming turfs of certain sizes. But also I believe the reproductive structures here are at the terminal ends of the branches and the pinnate structures, whereas in Corallina officinalis, I believe they're found throughout. You have your crustose coralline algae, like I described before. Generally, we don't identify these in the field. They're too morphologically similar. And again, you have to really either know them very well, but if you're going to do, for instance, scientific research with them, you have to identify them with genetic markers. Same with marrow. Generally, you're going to need a DNA sample to look for genetic markers and identify that species. But if you get a good eye for it, you can sometimes distinguish pretty easily. And then here's an example of our freshicostose algae. So there's a few different species that are common, Hildebrandia, or genre that are common, Hildebrandia or Peysonelia. It's very hard to identify to the actual species level, but some of these crusts can also be the sporophyte stage of the foliose gametophyte species like Mastocarpus or Gymnogongrus. So they have a heteromorphic, some species that have a heteromorphic life cycle actually, you know, might look like a Hildebrandia or a Peysonelia. And then here's an example of a brown encrusting seaweed, and that's Ralsia verrucosa. Here, found on limpets. Okay, so I'm going to talk about, let me see if I can hide this a little bit. For the reds, I'm going to get into more of the leafy reds or the bladed reds. So this is very distinctive here, Dilcia carnosa, generally found in the subtidal environment. It's big, oval in shape or ovate, and it has very tough leathery blades. So you can't really confuse it with too much just because of the texture. It's very elastic in texture. Palmaria palmata is very common in the subtitle again. This guy divides like in a forked fashion, but it's a blade. It is leathery and slippery, but thinner than Dilcia carnosa. It can't really be confused. And it's often found on kelp types. So it'll be one of the dominant species found on kelp types, forming that sort of subcanopy, if you will. And the kelp here would be Cuvee or laminary hyperborea that is found on, not any of the others. This one is very lovely. So this species of red now has a midrib you can see here, and small ribs coming off the side. Looks like an oak leaf. I believe that's called is it called sea oak? This is a common name. It's pretty flexible, has a toothed margin on the edge of the blade. And this is, again, also often found on kelp types in the subtidal environment. This one is potentially could be confused with Delaceria sanguinea. The younger individuals of Delaceria can sort of look like Phycodrus, but Delaceria, once it gets a little bit older, starts looking really ratty and stuff, and it's pretty hard to confuse. The difference, again, if you look at the midrib, is going to be there's no side branching off the midrib here. It's just one straight ovate frond or blade. Okay, Calophyllus laciniata is now called Metacalophyllus. I think it's still called Calophyllus in the C search book, though. To double check. This is a very beautiful one. It can be found as an epiphyte, like growing on other seaweeds or on the bedrock. It has kind of a fan shape to it, and it branches into wedges at the end right here. See? And then we have our eyelash weeds. So you'll note that this guy, the holdfasts haven't really been very apparent in the past few slides. So here we have Dilcia carnosa has an ovate or a discoid holdfast here. This guy has a very small discoid holdfast. But once we get to some of the eyelash weeds, you'll see they have root-like holdfasts, almost like kelps do. And this probably just helps it secure itself to more diverse types of substrates. The eyelash weed has a very long oval blade, and then it gets its name from the tassel-like growths on the edge of the blade. And there's two species of eyelash weeds, one that's big and one that's skinnier. So ciliata is going to be fatter, and jubata is going to be thinner and more branched. Okay, you guys might be very familiar with Chondrus crispus. This guy is found in the subtidal and in rock pools, but it can be confused with many subtidal species. So you have to be pretty clear. Like, so here's an example of another subtidal species, Rhodamenia pseudopalmata. And you can just get confused by the nature of the branching and kind of the flat branch structure at the distal end of these fronds. Yeah, the best way to ID it's going to be to bring it out of the water, I believe, before you, and then just double check before you submit your records to Seaweed Search. We also have Mastocarpus stellatus. The way you can distinguish between these two species is the edges of the fronds are going to be thicker, like there's a thick margin to it, whereas Chondrus crispus should be evenly thick across the frond. And then these guys have a really interesting variation in the 3 life history stages. So again, for Mastocarpus and Gymnogongus, there's going to be a crustose stage, but the reproductive structures are also very distinct on the blade. So if you find, I think it's going to be the carposporophyte is very, very bumpy, and that can help you identify the species. Mastocarpus and Gymnogongus are going to be more common in the intertidal, I believe. Okay, so another one that's found in the intertidal and also in the subtidal is Odenthalia dentata. This guy on paper looks like some of the browns that we might see, but it's pretty definitively red when you're in the field. This guy's regularly branched, has a very narrow midrib, and then the branching is going to be alternate on each side. It cannot be confused with coxcomb or the Placamium species, and I'm presenting them as species because there are likely multiple species of Placamium. They're kind of grouped into the Cartilaginum species name, but that's a name that's used from Antarctica to Hawaii to Ireland. I'm not entirely certain that's correct. So the coxcomb just identify it to genre, and it's very distinct types of branching right here. And something that maybe, but you shouldn't be confused, but does look like it from afar, is going to be Membranoptera alata. And the way I always think about this one is that it does look like fly wings or something like that. So the midrib is very distinct, very small frond margins, and this can be a common epiphyte on kelp, but also found on the benthic habitat in the subtidal and in rock pool habitats. Okay, another finely branched one like Placamium, but very distinct branching type is going to be Tylota gunneri. And this has opposite branches, distichous opposite branching. And it can go to three levels, right? So you have your main branching, then you have your side branching, and these have little branchlets off the side margins. It's pretty distinct, and it has a discoid holdfast right here, mostly going to be in the subtidal. And then we have a non-native Asparagopsis armata. This is very distinct. I think it kind of grows like a strawberry, so rhizoidal, and that would be the sporophyte stage here. Some people describe this morphology as arrow-like. I think it looks like an asparagus, actually, hence the name, maybe. And then the tetrasporophyte, the gametophytes look like this. The tetrasporophytes look like little cotton balls on the seafloor. So if you were to go out to Salt Hill, like in the winter time, and if you walk into the water a little bit and look down, you'll see these little brown tufts. And those are actually the tetrasporophyte just kind of rolling around. It's kind of free living. And I think this is how it disperses so widely and is so successful. Okay, so I'm going to talk about the browns after, oh no, I'm going to do more reds. Okay. So another intertidal species that you'll be probably very familiar with, you always see it in their intertidal, is somewhat iridescent. There's a couple different species, so you have to collect it and bring it back to, you can use this book to identify it, or a more complex key. It's going to be Osmundia. Maybe this is called sea oak, actually. It has a very distinct smell to it. It's very pretty. And it's going to be largely intertidal. The more sheltered the habitat, and it's going to be on rocky shores more often, the more sheltered the habitat, the bigger it actually gets. So when you get to more exposed sites, it kind of grows in very short turfs. This could potentially be confused with Gelidium. This is a flattened paddle spoon-like branching species, and it also has some creeping stolons. It might be a pseudoperennial, and what that means is some algae are annuals and some are perennial. And a pseudoperennial would just keep its hold fast year after year after year and only grow blades out seasonally. So when there's more light availability in the subtidal environment, potentially. Okay, these guys are pretty distinct, but I'm going to present two that are hard to distinguish just without touching or from the eye. So within their reds, they're fairly distinct. I guess you could potentially confuse this for a brown because it's such a dark shade, but it's a red. The Polyetes rotunda has a very discoid holdfast. It's forked, so dichotomous branching, and the fronds are circular. Forcillaria lumbricatus is similar branching, still has circular fronds. The ends of it are a bit flattened. So that's a one way to distinguish. But the easiest way to distinguish is when you're dieting. These are going to be mostly subtitle. When you're dieting, you kind of want to reach down and see what the holdfast feels like. It has a claw-like holdfast versus a discoid holdfast. And again, that's going to be handier for living in more diverse kind of substrates. Discoid would only really fuse to rock, whereas the claw-like might help it establish in sand or rubble. Our jellyweeds, this guy, here's a kind of a bad picture of it. This is a Cynia species. There's 3 different species. They're pretty hard to distinguish. So you generally just identify to the genre. They have circular branching. They're turgid, so they're water-filled. They look like a little bit of a balloon animal. And they're dichotomously branched. Another jellyweed is Lomantaria species. I really like this. This looks more like a balloon animal because it has those pinchings at the branch junctions. Again, circular branching, turgid, but very distinct delineation between, you know, where the branching is. And then we have a lot of turfs. So people will talk about turf algae in literature and also more in popular science now because it's such a it's a stable alternate state for kelp forests in a lot of places in the world, and also for coral reefs. And turfs are just smaller organizations of multiple species that generally proliferate either on sediment or on bedrock. And if it's a very highly disturbed environment or even just in a happy, healthy ecosystem, they're very common in the understory. So this is an example of some turf species, Rotothamniella floridula, that you might see covering sedimenty bedrock. Okay, now I'm going to move on to the browns. And I'm going to look at the chat really quick. Oh, I can't. Anyways, if you guys have any questions, you know, just put them up in the chat and we can talk about them later, or you can try and shout them out now. I don't know how Tony and Rory feel about that. Okay, so I'm going to start with the rockweeds with the brown algae. Huh?
I think shout away because sometimes they don't make sense when we get to the end.
Yeah. So yeah, just shout away if you feel like you have a question while I'm speeding through all these different species. Okay, I mentioned Ascophyllum nodosum or knotted egg-rack when I was talking about the zonation. This is going to be a very common, well, it's a very common species in Ireland. That is very common in sheltered shorelines, dominates sheltered shorelines. This is multi-branched, very distinctive because of this egg-shaped nematocyst, and it's very leathery, very strap-like. That is very closely related to a lot of our other rockweeds, but not as distinct, maybe more distinct than them. But these are actually fairly easy to tell apart. So there's 4 different rockweeds in this area, and I'm presenting 3 to you. You have Fucus spiralis. This has a nematocist or bladder at the end. Or actually, that's just a reproductive structure, sorry, at the end of the branch. You can see that this has a midrib within the frond as well. And it just spirals, and that's going to be how you identify it the easiest. I think it can be somewhat confused with gyri, but gyri, I believe, is still less common than the other three species. Fucus vesiculosus is very distinct. It has these nematocysts within the frond around the midrib here throughout the frond, not just the reproductive structure at the end. And then you have Fucus serratus, and this has a serrated edge to the blade. This is very common. And then you can see its reproductive structures here are at the distal end of the blade, but they're not swollen like they are in Fucus spiralis. Another brown, more subtitle, is going to be Bifurcaria bifurcata. This has forking branches, so dichotomous branching. It's cylindrical, and the end branches become very, very twisted, which is going to be a good cue for identification. Now, if you were just to go by the species description, you can see that this could be confused with a lot of reds, but it's pretty ochre in color, so it should be easy to identify in the subtitle. At the top shoreline, on our rocky shorelines, and also in some of the sheltered habitats, you'll have Pelvicia caniculata. Forms tufts up here. This guy actually I think it can survive up to 10 hours out of water. So it gets really desiccated and then it rehydrates itself at high tide. Um, and then we have our Araucarias and our Gongolarias. So these are the, shoot, what's that? Oh, our rainbow racks. And so they're pretty distinctive because they have this iridescent coloration to them. But they're hard to identify from each other. So Gongolaria baccata, also called bushy berry rack, has these pea-sized vesicles within the frond or the branches. I'd say branching is a better way. So the nematocysts are going to be your identification tool here. Gongolaria noticalis, the rainbow rack is iridescent, and the vesicles are often hard to kind of decipher or see easily. These are both the bigger of these three racks, and then you have your bushy rainbow rack, very iridescent, has a cylindrical axis, but it's often very hard to distinguish. It's just a bit smaller than the other two species, should help you. If you have a question about what species is going to be, it's easier just to collect it and bring it back to the shore for to key out. Now, you could kind of confuse this with Hylidris silicosa, but the Hylidris is very, very bushy and big. And it's, I'm not going to say more common, but I've noticed it a little bit more in diving than these other ones. You can see the Gongolarias and the Araucarias in the shallow subtitle and low intertidal sometimes as well.
Actually, Hylidris
And
is also called sea oak, which we've
is
had
it?
already. Yeah, that's why I don't use sea oak as a description. That's a common name.
Right, right. Common names are rubbish. Okay, so this is our non-native brown. We have this in California. It's pretty much, I think it's pretty much it's all over the world, pin. Um, it's found in the subtidal, often in disturbed areas, seasonally. Really, really does well in the summer season. It's very bushy, but it's got alternate pinnate branching, unlike the Hylidris, and it's not iridescent, so you can easily distinguish from the Gongolarias. It has spiny blades as well, like some of the tropical seaweeds that it looks like or shares habitats with. And the bladders are very, very round. This guy can be really long too, so he can grow in deeper waters. Often you can see him kind of growing on other seaweeds, and they get up to be maybe 4 meters or longer, depending on the habitat. This is a picture from a shallow habitat from seaweed.ie. Okay, here's our kelps. And I've drawn these out because I think that it kind of highlights the morphology a little bit better than just a picture. So our oarweed here, Laminaria digitata, has a very flexible stipe. And it has haptera, so a claw-like holdfast. And its blade kind of breaks up into thin fingers because it's found in the low intertidal zone predominantly. It can be subtidal sometimes, but it's generally in the low intertidal where there's a lot more water action. So that's going to tear and break up blades more. We also have Laminaria hyperborea. This is your subtidal dominant. It can be also found in the low intertidal. And the easiest way to distinguish between these two species in most habitats is it has a rough stipe with a bunch of stuff growing on it. And it lives a lot longer, so it'll be a lot bigger, but the stipe is also rigid. And the holdfasts are going to be larger and have a lot more colonizing organisms on them. And we have our somewhat non-native species. So this is Laminaria ochrolouca or golden kelp. I think we still have only seen it in one habitat in Ireland, and that's on Belmullet. But it is getting more and more common in England, in South England, generally kind of occupying more disturbed habitats or man-made habitats like harbors in the same way that wakame does. But in Belmullet, we found it just kind of dominating one cove. So kind of an interesting species. We haven't really seen it anywhere else in the subtitle, but something to keep an eye out for. So like digitata, it has nothing colonizing the stipe here, but it does have a rigid stipe. The color of the blade is much more golden than what you'd see in hyperborea, but young laminaria hyperborea that are under 5 years age can also have that fresh blade and maybe cleaner stipes. And then if you look at the hold fast, the claw-like hold fast, in hyperborea, it's kind of mounding. Every year, new claws kind of form and reach down to the bottom and then get colonized. But ochrolouca and digitata, those guys really have more planar claw-like holdfast. They really spread out and flat, like a flat plate. Okay, other kelps are going to be more distinctive. So you have your sugar kelp here, Saccharina latissima. This has a midrib and it has a very rugose or ruffled blade. And this can be up to 2 years. So the older individuals are going to look more weathered. The younger individuals can actually have a very long stipe in some sheltered habitats like, uh, shoot, where do where do we dive on the boat in the lock? Lock, not Loch
Lock
Eyen,
line.
but no, not Loch Eyen. It's north. It's in sugar. I can't think of it. I can't. Brain dead. Um,
Strangford,
not Strangford in
Axvillian,
Galway.
in Galway.
You know where we launched the boat, the rib? You can dive on that boat wreck or go to the wall.
Oh, you mean Killary?
Yeah, fjord, Killary Fjord. Fjord. Uh, so yeah, but I've seen Saccharina latissima there with very long stipes for the species, and but the blade is still ruffled. So it's just like depending on the habitat and the amount of environmental action, kind of the energy in the habitat, it can change its morphology a little bit, and then also the age kind of changes the way it looks. Um, anyways, it's kind of a cool species in that sense. Porabellos is right here. This is Saccharisa polyshetes. He's very distinctive, right? The holdfast kind of looks like a dog toy. Then it has these ruffled margins, which produce the gametophytes or the gametes that then form gametophytes in this species. Um, and the tops of the blade here are going to be very V-shaped. So if you're just swimming over a canopy, that's how you identify that species in comparison to, for instance, Laminaria hyperborea. And then we have our non-native wakame or Undaria pinnatophyta, which I think still to this date has really only been found in harbors, but it's pretty common across Ireland. at least in the eastern and southern region. Okay, here's more of our intertidal browns. I'm going really long. Here's Copalmenia peregrina. It's can be in the subtitle, but it's mostly intertidal. It's a very sac-like appearance, and the way you're going to distinguish it between its fellow species that is also sac-like is it's going to be very soft. Lithisia marina is also found in the intertidal, but it's very hard and crunchy. So you always want to kind of test texture when you're identifying species and you don't want to collect them. We also have sausage weed, so Asparacoccus species. Bullosa species is a hollow tube. It's going to be your bigger sausage weed, and Fistulosa is going to be your thinner sausage weed. Chordophyllum is a very common brown alga, so it's cylindrical, can be 10 meters long. It's a very hairy appearance, and you'll often find it in habitats like Killary Fjord, or maybe just a little bit shallower than a kelp forest if you're on a more exposed coastline. Pimenthalia elongata looks like little buttons here before it starts growing its larger thallus. So you'll see these in the intertidal, shallow subtidal. It's called spaghetti weed. And it forms a flat belt, dichotomously branched. You generally see those buttons in the winter. So from December to March, I believe, is the timeline. Okay, here's some smaller guys. Here's Dictyopteris polypodoides. And this looks a bit like fucus, but it has a very apparent midrib with no nematocysts or apparent reproductive structures. And it's generally going to be in the subtitle. We also have Dictyota species are very similar in size and coloration, but they have no midrib. There's a lot of these different species, and they're pretty interesting from a chemical ecology perspective. But generally, you're going to just identify to the genre with that one. And then you have Teonia atomaria. This guy looks a little bit like Dictyota, but it has these hair-like rings as you move up the frond. That's going to be how you identify it from it. Also looks more frayed and broken up. Other browns are going to be Corda linearis. This is a long whip-like alga. Believe it has a discoid holdfast. Could be up to 70 centimeters long and could be confused with the Desmorosiales, but it's pretty distinct in that it's got clean fronds. Desmorosia aculeata is about 1 meter in length. It has alternate branching with these hair-like fine branching that resemble hair. Well, hair-like. Has a bulbose holdfast. So it could be mistaken for viridis, which is pictured here growing on a mussel shell, but viridis has a flattened holdfast. So always important to kind of reach down and just double check that structure before you identify it or report it. And then you have your Desmorosia ligulata. This is very distinct in that it has very flattened branches and multiple levels of branching. So up to 3, I believe. And the bulbous holdfast. Okay, I believe this is our last brown, and this is Mesogloea vermiculata. This is gelatinous, so I think it could be easy to confuse with some of the red jellyweeds. But it's going to be more ochre in color. And you just have to look at this random branching pattern that it has. And you can see it does look a little bit like a jellyweed when you have an in situ picture. Okay, I'm getting close to the end. So here's our green seaweeds. These guys are like the coral and algae in that they're very hard to just identify. So you have your ulva species. They can be confused. These are your sea lettuces. They can be confused with Prassiola species or Monostroma species and Porphyra, which is a red. They're generally found in the upper intertidal estuarine habitats and the deeper subtidal habitats. So you'll probably encounter them in the intertidal or an estuary. They're generally very ephemeral in that they grow when it's warmer and there's more light. Like for instance, here in California, we get big, I wouldn't say bloom, as in like with a bad context, but we get huge communities kind of washing up on the beach from June through August. And I believe it's similar, especially in estuaries in Ireland. You have dead man's fingers, Codium fragile. It's dichotomy branched. This is a siphonous alga. Has a very hairy appearance and is mostly intertidal. It can't really be confused with anything, but there are two subspecies, and one of them is thought to be more common. I generally just report Codium fragile, but Sea Search could tell us to do something somewhat different if they wanted to. And then we have Cladophora rupestris. This looks like Grinch fingers, I think. When the way it moves, it's very like green fur, maybe Oscar the Grouch. And this could be confused with Acrosophonia or Spongomorpha. But if you collect it and bring it in for the key, it should pretty much stand out, the Cladophora species. And then we've seen Codium bursa. Tony's especially seen Codium bursa. It's pretty rare in Ireland. I think it's only been seen in, I'm drawing a blank on the site name. For the Saperla
Kilcairn Bay
vermicularis.
and Mulroy Bay.
Yeah.
But there's a lot more records of it online than I thought.
Oh, really? Oh man, sorry, my kid's melting down. And then we have our seagrass. So this is a angiosperm that grows in more brackish and marine habitats, generally shallower. And Zostera marina, you might know it because it's an ecosystem engineer and it's got a lot of press on being a great system for carbon capture in the marine environment. And there's a lot of mapping that's been done in Ireland, yeah, both through the EPA and University of Galway. Okay. So I'm just gonna run through how you do some seaweed presses. I have a press here, but basically you don't need to have anything fancy. You just want to have somewhat nice paper, non-acidic paper that's absorbent, so watercolor paper. Generally, a paintbrush or something that you can use to manipulate the blades, especially for finely branched species that are like the ones pictured here. And then two boards, or like let's say a book and the floor, something hard to press the paper between, newspaper or other absorbent material, and then cloth squares or cheesecloth or a nappy liner or wax paper to kind of protect the seaweed when pressing it. So the way I like to do this, and the way I taught my students last summer, is I like to get a diverse array of species, so like reds, greens, and then the smaller browns, generally juvenile browns that are not going to be so tough and thick. You bring them home in seawater. You want to make sure that you don't have any contaminants in that water, so that the like might bleach the seaweeds out or somewhat. You're gonna want to try and get a cookie tray or some sort of maybe like a baking sheet, um, and place your paper in there. Place your seaweeds in the arrangement you want with a bit of water, and then you can float and kind of arrange the seaweeds as you like on the paper. Then you can lift your paper out of it, tilt the water off just enough to like enable quick drying, but keep the seaweed in place. And then again, go through it again with a paintbrush so that you can spread out the seaweed branches, that you want to really kind of capture the morphology and the branching structure with your pressings, so that you can kind of go back and look at them later. Don't want to have any kind of huge overlaps or thick stacks, because one, you won't be able to like go back and see if you did your species ID right really easily or not. But also it makes it more prone to molding when you have that kind of thick clumping. So here's just kind of a walkthrough. Basically, place a wooden board on a flat surface, cover with newspaper. This can be your absorbent pad. Place a watercolor paper with your seaweed on top of that. And then you're going to use cheesecloth for the thickly bladed seaweeds like kelps or maybe like Dilcia carnosa. You can use a nappy liner or wax paper for your thin species so they don't get stuck in that cheesecloth. And that's just going to protect that seaweed from getting picked up by the newspaper instead of the watercolor paper. So you lay those that over the seaweed, cover again with newspaper, and then repeat as much as you want for all of your seaweed pressings. Then you're going to put a flat board or maybe a book on top of this and make sure it's really, really weighted down. And you want to leave several days to dry. The most important thing for doing this in Ireland is that you do it in a dry, not humid location, and that you check on it at least daily until you're uh, satisfied with the pressing, because they do get very, especially even the thicker reds like Chondrus crispus can start building up this like thin veneer of mold. And so here's just some examples of some pressings from online. You can also do sun prints if you want. This was very common in Victorian era. And they can look really pretty as well, but they're not so great for looking they're not so great for uh creating a species record necessarily, or a print that can be used in a sort of a scientific context. So that's it for this presentation. This is just a nice infographic that one of our colleagues did for our kelp-res project. It's just a nice depiction of the diversity. You can see you have your Helidris, your laminary hyperbrea, all your epiphytes. Nice depiction of the diversity of seaweeds in the subtitle.
But they didn't label the Lysocalum perforatum, as Art pointed out.
I know. I mean, can only do so much, I guess.
Thank you very much. That was fantastic.
Sorry, that